Hey everyone, welcome back to our final lab of the semester, the Mammals Lab, Mammalia Lab 15. So this is where we are now. We're looking at the mammals. As you can see, there's a couple apomorphies that unite all of them. Down here, all right, you've got hair, mammary glands, middle ear, made up of three bones. You can see them all there, right? Big groupings, you're going to have this outgroup of the monotremes, right? They're the most out there in terms of still being mammals, but not like anything else you've seen. And then we've got the marsupials, and then we've got the eutheras. So you have thera, all right? That's going to include the marsupials, and then you have the euthera, which are going to be all your placental mammals. As we look at the mammals, apomorphies that are going to unite all of them are going to be this heterodont dentition, meaning that they're all going to have different types of teeth. If you remember looking at, say, like the crocodiles, they had that homodont dentition, so all of their teeth were these conical shapes, different sizes, but basically the same shape for catching fish. Now we're going to see many different types of teeth for different types of eating. Got a single bone for the lower jaw called the dentary. Middle ear is going to be composed of three bones, the stapes, the malleus, and the incus. A four-chambered heart with left aortic arch only. Right? That's going to be an important thing to remember because I'll get into that later. That's why it's different than some of the other ones. We now have this muscular diaphragm, which is how we get this negative pressure breathing. The presence of mammary glands. Mammals, mammary glands. Right? They're going to be different in all the different groupings, but generally that's going to be one of the big things. Endothermy, meaning that mammals control their own internal temperature. They're not subject to the outside world. And the presence of hair. Now, when we look at the dentition, heterodont teeth, right, meaning it's teeth of different shapes and sizes. This allows for more precise chewing. This is a general mammal dentition plan, right? So there's going to be a million variations of this, but generally speaking, you're going to have incisors for slicing stuff, canines, right? Premolars, and then molars. Now, like I said, depending on whether you're looking at the herbivores or the lagomorphs or the carnivores, some of them are going to have almost entirely just molars, or you'll only have incisors and then some molars. But like I said, this is just a generalized mammal dental plan. I'm going to let you kind of go more into depth on this. You can kind of study this slide. But basically what you want to remember is that there's been a reorganization of the jaw bones that had a profound impact on the middle ear and the hearing. So evolutionarily speaking, you can see how a lot of these bones have shifted and evolved over the millions of years. And they've got this nice little color-coded thing so you can see how different bones have shifted. And so single lower jaw bone is called the dentary. Middle ear bones passes vibration to the eardrum to the nervous system receptors and articulate, and the quadrate bones became incus and the malleus of the middle ear. But you're going to want to kind of study that a little bit more. The four-chambered heart, right? If you remember, this isn't the first time we've seen a four-chambered heart, but what you definitely want to take away from this is now we have a separation of the pulmonary and the systemic circuits. So the pulmonary with the lungs, and the systemic circuits, the heart stuff. So you've got lungs, all right, so your pulmonary action, which is going to be enriching the blood with oxygen, and then your heart circuits, which are going to be pumping it around the body. So this has evolved independently from the Archosaurian four-chambered heart, and they are not homologous to each other. The right arch has been lost. Right? Reptiles have both arches. Birds have a right arch. Mammals have just the left arch. And then if you've done any of the anatomy or physiology of humans or any other animals, or you will, what you want to remember, right? You've got high pressure going from the heart here. It gets pumped into the lungs, gets enriched with that oxygen, comes back into the heart, which then it gets pumped back out again. So you got nice high pressure, pushing to the rest of the body. These are going to be your arteries going from the heart to the body, and then your veins, which is going to be low pressure because you got big, thick arteries going into these tiny little capillaries. So picture like a big hose getting into much, much narrower hoses, little tiny tubes. But then once you get into those tiny tubes, then you're going from the tiny ones out to the big, thick ones, so there's going to be much lower pressure. And if you get into medicine, that's going to be your systolic and diastolic rate. You're taking blood pressure, you get certain numbers over another one. So that's the pressure coming out, pressure coming in. And so oxygen-rich blood goes to the body, gets used up in the muscles and the body systems, gets taken back to the heart, and then gets pumped back into the lungs to get reoxygenated, and the process continues. The diaphragm, right? This is that muscle right down here that's below your rib cage. Now we're going to have aids in breathing, increased metabolism, and negative pressure, which means that when your lungs inflate, it's not a muscle that's expanding your lungs. It's this diaphragm that's doing its thing and changing the shape of your rib cage and making that open up and then just kind of sucking the air in. Right? So instead of your lungs having muscles that make them bigger, they're just expanding the rib cage around them, causing negative pressure and that air to come rushing into your lungs. Which is why if you've ever been hit in your solar plexus or you have the wind knocked out of you, it's caused your diaphragm to spasm, and all of a sudden you can't draw air in until it kind of regains control. Kind of scary, but not uncommon. Mammary glands, right? Probably one of the most distinguishing features of mammals is this presence of mammary glands, right? And this is so that they can provide milk to nourish and feed the young, and they are simply modified sweat glands. They all have them, so all mammals are going to have them. It's one of the defining characteristics. Now, the monotremes, that's going to be your platypus and your echidnas, they secrete milk to the surface hairs, right? While most other mammals are going to possess nipples of some kind. So the monotremes, again, very much of this outlying group, and so they're really, when they're showing that these are just modified sweat glands, they're just kind of sweating out milk in certain spots, and it's going just to the surface hairs. Everything else gets nipples, and allows their young to be able to get all the nutrients they need until they can eat solid foods. And depending on what type of animal you're looking at, they can be very, very different. Like some of the marine mammals, their milk is so fatty, but if you live in the Arctic or live in these really cold waters, you need them to quickly build up that fat layer so they can protect themselves. Fun fact, camel milk is one of the closest to human mother's milk. So if you ever have a situation where there's a newborn that needs milk and you don't have any mothers and you can find a camel, you'll be okay. Another apomorphy is going to be the presence of hair and endothermy. So endothermy, the body heat is internally generated as a byproduct of metabolism. So the term regulators and conformers are more important for the lecture. That's why I'm throwing them in here. Doesn't really talk about it in your lab manual. But just remember that for endothermy, the organism is regulating their own body temperature. They've got various set conditions inside of the bodies, and they want to make sure that they're in that temperature range, and they're going to do everything they can to maintain that. Whereas ectothermy, these are conformers, meaning that their body temperature is going to be subject to whatever the ambient conditions are outside. Think of reptiles who sun themselves, so they go lay out on a warm rock as they're basically just trying to get their body warmed up so it can start doing all of its processes. Now, what you have to remember about the ectothermy, so these conformers, they have to lay out in the sun to warm up, but if they're out there too long, they're going to overheat, so they've got to get back into the shade. So it's just kind of this constant little dance of balancing your body temperature out, whereas the endothermy, or the regulators, aren't so much subject to that. Now, if you think about yourself, human body temperature, roughly 37 Celsius or 98.6 Fahrenheit, depending on which one you're looking at. All right, that's 98. We'll take Fahrenheit, 98.6. If you bump that up by 2 degrees, you're into fever temperature range, and if you go up by 3 or 4 degrees more than that, you're talking a severe fever, and like 108, most people are going to die. and that's only a couple of degrees difference. Right? So your bodies are very good at trying to regulate their own temperature, and that's why taking temperature checks are a really good indication of something's going wrong. Now, if you don't know, the reason that your body generates a fever to fight infection is because proteins will start to denature at certain temperatures. And the idea is that if you can raise your internal temperature high enough to denature, to break down whatever is infecting you, then you can fight it off. Hopefully, you'll be able to do that before your own body processes start to denature. So, like 104, you can kill off a lot of bacteria. 108, you're going to die yourself. Now, the way they can achieve some of this regulation is body hair. So the bodies are going to be covered with hair for insulating a lot of the mammals. So you can see, like, with this yak or the big fluffy dogs. In other cases, the hair is not so much as important as the blubber. Like, for instance, with marine mammals, for like seals and whales and whatnot, the blubber is the most important part of keeping them protected. So they do have the fur on them, and that it does help. But if you look at a whale, whales aren't known for being hairy; they're known for being blubber rich. So they have the hair, but it's just not that important for them in terms of thermal regulation. Now we're going to look at some of the taxon of these different groups of mammals. On the most extreme outgroup, we've got the monotremes, right? And these are going to be your echidnas and the duck-billed platypus. There's only 3 living species that inhabit Australia. 3 living species total, and they inhabit Australia and New Guinea. And that's going to be the duck-billed platypus and the spiny echidnas. Now, what you want to remember is they are oviparous, which means they lay eggs, but this is an ancestral trait. It is not an apomorphy for this group. Lots and lots of things lay eggs. Birds lay eggs. Mammals lay eggs. Birds lay eggs. The reptiles lay eggs. So laying eggs does not make them special. What makes them special is the loss of teeth in the adults. So they just don't have teeth. Unfortunately, I can't show you. We are one of the few campuses in the country. There's only like 2 or 3 other universities in the country that have an actual preserved specimen of a duck-billed platypus that you could touch and look at. I think it's us. It's like Stanford and Yale, some places, but most universities cannot have them. Australia very tightly controls what happens to the duck-billed platypus and the spiny echidnas because they're a national treasure. Think of China with their regulation of pandas. Same thing. There are no currently living, to the best of my knowledge, and I've looked this up as much as I can. There are no duck-billed platypuses alive outside of Australia. They just don't let them out, and for pretty good reasons. If you're ever having a bad day, look up videos of duck-billed platypus, especially baby ones. They are adorable, and I'll probably be showing you some of those during the activities walkthrough. Now, the rest of the mammals are going to fall into this group called the Thera, right, which just means wild beasts. But they're going to be united in having, so they're the sister taxon to the monotremes, and this is going to be containing your marsupials and your placental mammals, right? It's just Greek for wild beast, the Thera. They are now viviparous, right? Before you had oviparous, ovi is egg. Now we've got viviparous, meaning live, right? So the young develops in the mother's uterus, and then they're birthed live. There's now placentation between the female and the embryo, and that just means it's going to be a placenta, which is the union of uterine and fetal tissue, allows nutrient uptakes, waste elimination, gas exchange, blood supply. Basically, it's placenta. It's this little cocoon of goodness that helps the baby grow. Now we're going to also start to see an expansion of gray matter in the brain. Most instances, we're going to see this highly folded neopallium, the cerebrum, associated with learning. And now we're starting to see 2 excretory openings. We now have an anus and a urethra. Before that, you were always going to be the cloaca. So the birds have the cloaca, like a reptile. Everything else had a cloaca, which is just one exit for the solids and liquids. And now we're starting to differentiate. You've got anus for solids and urethra for liquids. And the presence of the pinnae, right, which is just these external ear flaps. And if you were to ever get into the marine mammals, which unfortunately the class no longer happens, but the pinnipeds are, pinniped, it's just, they got ears. One of the defining things of true seals. Okay, now we're going to look at the marsupials. Epimorphies for the marsupials are going to see the marsupium, right? That is the ventral pouch which the young are carried. It's really important for their development. The choriovitelline placenta, right? That's just formed from the chorion and the the yolk sac for nutrients, right? What you need to remember about the marsupials, they have a very short gestation period. So between conception and quote unquote birth, it's very short, like a month. And you compare that to humans, 9 months, elephants, 22 months, right? But when they're born in this fetal state, meaning that they're still fetuses, right? And so I'll show a video during the activities portion, but they're very tiny. They'll crawl out, being born, and they'll crawl into the marsupium. They'll latch onto one of the nipples, and then they'll finish their development there for, like, some of the kangaroos, it's like 34 more weeks before they'll leave the pouch. And so that's what I mean by a very short gestation period. Marsupials are primarily found in Australia and New Guinea. However, that's not the only place in the world they're found. In America, we've got the Virginia opossum, and it is an opossum. First, they're naturally resistant to rabies, so you don't want to kick them out of the air. They're not like the regular trash pandas you see. So first, they're adorable, and two, they're naturally resistant to rabies, and they happen to eat ticks like candy. Right? So don't confuse them with a possum, though, because this is a possum, big fluffy tail found in Australia. This is America's opossum, both marsupials, right? But completely different animals. So again, they're just adorable. They're found locally in our area. Right underneath my apartment complex, we had a mother, and she had 8 little ones, and sometimes you'll see them crawling on her, they'll latch onto her back, or if they panic, they'll all cram into her little pouch, and so you'll see this mama opossum walking around with 8 little tails coming out of her stomach, and it's really cool and cute. The Tasmanian devil was a real thing. Right? Does not look like this, right? No, but they do look like this. Right? And why am I telling you about them? Because one, they're cool, but two, they have been subject to this thing called the devil facial tumor disease, and it was one of the first times that I'm aware of, if I'm not mistaken ever, that they saw cancer being transmitted as a contagious disease. They figured this out because their population of this thing was being decimated, 95% decline in 20 years, and they all have these crazy facial tumors that were growing. But unlike normal cancerous tumors that would grow, which would have similar genetic composition to the person who has it, all these tumors share very similar, almost the same genetic composition, different from the host. It turns out because Tasmanian devils, they got the reputation like this guy because they do like to fight, and they would be ripping and tearing each other's faces, and they would be basically open wounds, get these cancer cells embedded in them, and new cancer would grow, and it would eventually kill them. Not very cool. Now what we have here is the thylacine, also known as the Tasmanian wolf or the Tasmanian tiger. And so why I'm showing you this is because this is the largest of the known carnivorous marsupials. More importantly, it's showing that an ecological niche needed to be filled. So other parts of the world that you have large carnivorous cats like lions and tigers, other parts you have large carnivorous canids like the wolves and the coyotes, but you don't have that in Australia. You've got marsupials. And so this thylacine evolved to fill that large carnivorous ecological niche. So even though it looks similar to a tiger or a wolf, there is no there's no evolutionary connection to them outside of, way back on that evolutionary tree. They just have to evolve to fill the ecological niche, and, they will look similar. You're going to see something similar to that in the activity section when you'll see a marsupial mouse. You can see, marsupials, no connection to their placental counterparts other than that last common ancestor, however many millions of years ago. Moving on from there, we have got the koalas. Oftentimes erroneously referred to as koala bears. Again, they're marsupials, so there is no connection to the bears. Right? They are adorable, but unfortunately, they are just dumb as dirt, and it's really sad. But they're being described as being painfully stupid. They eat only eucalyptus, which has very low nutritional value and is slightly toxic. And so they have, you know, one of their biggest threats is chlamydia. Now, I used to go on a rant about koalas, but I'm not going to do that this time because their numbers have been decimated. If you don't remember life before COVID-19, one of the biggest stories was that Australia was basically on fire, like a whole large portion of it was. And hundreds of millions of animals were dying, including threatened koala populations. And because they're not very bright, they would be up in their trees and they couldn't leave. I mean, when I say they're painfully stupid, they can eat eucalyptus leaves off of a branch. But if you take those leaves and you strip them off of the branch and hand them to them, they don't know what to do with them. And their brains, there is the smallest brain to skull ratio of any mammal. Right? Their brains only occupy 60% of their skull. They're smooth, and the rest of the area is filled with fluid. So when they fall out of the trees, they don't get any more brain damage than they need to. But because of this low ability to learn, when the fires came, they couldn't do anything, and they would just basically be in the trees and get cooked out of the trees. It's really sad, and so they're trying to bring their populations back, and they're not extinct yet, but some populations might have been irreparably damaged in the massive fires. Cool thing, if you can see here, they have 5 digits on their fingers like most other things do, but they've got basically 2 opposable thumbs and 3 fingers. So we've got 1 thumb, 4 fingers. They've got essentially 2 thumbs and 3 fingers. Just helps them kind of grip onto things. Now, saying the koalas are adorable, but don't get them wet because they lose their adorableness. They're kind of scary. Same thing if you look at a black bear. Black bears are adorable until you shave them. There's a medical reason why this bear got shaved. That's a black bear when it's lost all of its fur. Moving on. So, we've looked at the monotremes, right? They were the outlying group of mammals that lay eggs still, but they've lost their teeth. Now we've got the marsupials over here, right? They got the marsupium, they got that pouch. Now everything else that you're going to be familiar with is going to be the placental mammals, right? Those are going to be broken into 3 smaller groups than the placentals. Right? So you got your Xenotheras, your Boreotheras, and your Afrotheras, right? To look more in depth. So now we're going to look more at the placental mammals. Right? They've got this prolonged embryonic development, and the embryo is nourished through the chorioallantoic placenta, which is formed between the chorion and the allantois from the egg development. They've got a corpus callosum, right, which is this connection between the 2 halves of the cerebrum, and it facilitates interhemispheric communications, a lot more brain activity going on. And the big groupings are, like I say, are going to be the Xenothera, the Boreotheras, and the Afrotheras. So the Xenotheras, so the first subgroup we're going to look at are going to be the Xenothera, formerly the Edentatas, because they were originally classified as not having teeth, but that's not true because not all of them are without teeth. So we've got anteaters down here, all right? One of the big groups of them are going to be the vermilinguas, meaning worm tongue. And if you've ever seen an anteater, its tongue is very worm-like because it's putting its tongue into anthills and termite mounds and whatnot. Right? So there's lots and lots and lots of those. They're really kind of cool. Sloths, one of my favorite animals of all time. Right? They're just awesome. And then the armadillos. Right? These are native to the American South. Sorry, they can be found in the American South, like Texas and all that area. Fun fact, they can carry leprosy. And we'll get more in the lab, but their skin, so you have like the reptiles with the turtles, where they have that big test, which is part of their skeleton. These guys, their skin is embedded with bone, right? But it's not part of their skeletal structure. They've got a regular skeleton inside. They just happen to have bony plates in their skin. And these bony plates are so strong that it is not uncommon to hear, you check the news sources out of Texas every year or so, and somebody's going to get shot in the face or shot because they're trying to shoot one with a shotgun, and the shot pellets ricochet off of the armadillo and hit the hunter. So take that for what you will, but I'll try and just find a video because they pop into this little ball, and they look like a little soccer ball, and it's quick, it's a pop, right? So they get scared, and they've got this little armored ball something's gonna have a hard time penetrating into. Moving on from there, we've got the Boreotheras, and what does that mean? Well, the Boreotheras are going to have a northern or boreal distribution, and these are going to be the big groups of things like you got your moles, so your Lipophylias, your carnivores, carnivora, primates, right? That's apes, monkeys, and us. In case you didn't know, we're primates. Chiropteras are going to be your bats, the rodentids, right? Your rodents. That's one of the biggest groups that are out there. And then lagomorphs, hares, pikas, and rabbits. Now, a lot of people think that rabbits and stuff are rodents. They're not. Two completely different groups, and you'll see why why they're totally different. So some examples, some other ones are going to be your Perissodactyla, right? So your odd-toed ungulates, rhinos, horses, and tapirs, and then your Cetariodactyla, even-toed ungulates, which are going to be all the other hoofed animals, and whales. Right? We classify whales in this group because evolutionarily, whales used to have a lot of legs, and even still, whales are going to have vestigial hind legs that you can't see, but they're sometimes embedded into their body still. But that's going to be things like your giraffes and your cows and other stuff, right? But yes, even the orcas, who ask me why I don't like them later, but world's biggest dolphins, fall into this category. And then the last group is going to be the Afrotheras, which are the African ones, originating in Africa. So these are going to be things like your hyrax, which we would have been looking at if we went to the zoo, your elephants, and then the sirenias. So manatees and dugongs. You got your proboscidae, which elephants got the big, pronounced proboscis, the nose, and the hyrax. So wish you guys could see these. If you look at the skull and the feet on a hyrax, their closest relatives are in fact elephants and the manatees, not the rodents, not the lagomorphs. Because when you look at them, they actually got their feet like little hooves, and they got these wicked, evil-looking skulls. But whatever. Okay, so that concludes the lecture for the mammals. Make sure you follow the link to the lab activities video. Do the write-up as normal. Please do your best to find and include a genus and species. If nothing's provided, you are more than welcome to look up a genus and species for something that interests you that we've covered. But make sure you aren't just describing a broad taxon of organisms, that you use it, the write-up to describe something specific. If it's just a picture of something, then you can say no genus and species given, but it's not ideal. And make sure you click through all the material on Blackboard for the lab activity. That way you get credit for that. It's easy points that you don't want to be throwing away. Exam 6 should be up on Wednesday, so same as before. You're going to have 24. It'll be open for 24 hours. Once you start it, you're going to have 30 minutes. You have one shot, so make sure that you're ready to sit down, because once you start it, you cannot reopen it. It is not open notes, but not open internet, so do your best to study for it and be ready. Again, you're only going to have 30 minutes, so if you try and look up every answer, you're not going to do very well. And then don't wait until the last minute if humanly possible, because tech problems happen, and if you wait till the last hour and something goes wrong, then you may not. Anyway, so don't wait till the last minute if humanly possible. Virtual field trip write-up due May 7th at 5 p.m. Make sure you read the instructions. Don't forget to include the URL for each of the webcams you've watched, as well as the time that you were logged into there. Genus and species are the best if you can identify them. Keep in mind, if the webcam only identifies the common name of the organism you're looking at, you can look up the genus at least. Most likely, though, if it's this is a rate something or other, you'll be able to get the actual genus and species. So that shouldn't be an issue. If you are interested in doing an undergraduate teaching thing, so the UTAs, which normally you would have been seeing helping out in the lab every week, contact Dr. Reckdale and or myself. You should have a B+ or higher in the course. However, as long as you pass, meaning you get a C, you're really enthusiastic about it, let me know. Right? If I think you'd be a good person, I'll definitely give you a recommendation for it. It's worth 1 unit of Bio 299 credits. If you need, hey, I need 1 more unit to fill my schedule next semester, this is a great one. And frankly, it looks good on resumes, saying that as an undergrad you were part of you're one of the teaching assistants for a lab, if you're trying to get into a research field, or just, it looks good on a resume, if nothing else. Evaluations, there should be, not everyone gets one, I think, but be sure to fill out evaluations online whenever you're going to look at your final grades. Please be honest when you fill it out. I do look at them, and if you want, your grades, I don't see them until after your grades are done, so it cannot have any effect on what your grade is, and they're completely anonymous. So please either fill out the evaluation or send me an email, something that I could do to do better, or what needs to be improved. I know the lab manual is not very good. We've been working on that, so they say, for the last several years. So don't critique me on the quality of the manual. If you have been interested in anything we've talked about this semester, here are a list of some of the classes that are offered at the university that are entire semesters based just on what we covered in the course of a week. I can personally tell you, I would recommend 554, this microbes one with Dr. Lipson. Kind of challenging, but super interesting as to how much our world is completely controlled by microbes. I just finished taking 514 with Dr. Matt Edwards, the biology of the algae class. Again, super interesting. If you're at all interested in oceanography and marine biology, it's going to be very valuable. And then there's just a whole lot of other ones going on. Unfortunately, marine mammals, I don't think is being taught anymore after Dr. Berta retired, which is a shame because she was amazing. And the animal behaviors class with Dr. Clark, if you can get into it, I know it's always waitlisted, I would highly recommend. It's challenging, but probably one of the most enjoyable classes I've taken here at this university. So this concludes Bio 204 Lab. Thank you very much. You guys it was a great semester, all things considering. Really want to thank you for your patience in dealing with the chaotic nature of the semester. This is the first pandemic I've ever dealt with, hopefully the last one. And you guys have all been shown a lot of patience while we got everything kind of switched to an online format over the course of like a week. Please contact me after you submit your virtual field trip write-up if you wish to discuss final grades. Want to know why you're getting something or wondering how you're doing. But wait till after that's been submitted so I have time to compile the grades and I can tell you what needs, where you're at. Good luck with the rest of your classes. I know that it's going to be a little chaotic in the next week or two with finals coming up. And last but not least, please stay safe, and hopefully I'll see you guys around campus. Take care, and again, thanks for a great semester.